We'll have the last speaker of our first session today, Mike Halko from Harvard Medical School. And he is also a very interesting TMS target. The cerebellum will talk about more in his presentation. Hand it over to you, Mark.
Thanks so much, Alex, for inviting me. And also, it's a pleasure to sort of see a bunch of familiar faces in the group. It's always fun to see friends. So I want to thank Joel for sort of leading me in, and hopefully he doesn't get too upset about me talking about his work too. Just real quick, the whole point of this is to sort of talk about work that we've been doing here at Beth Israel and now going to be at McLean. So we're really trying to look at how do you sort of manipulate the cerebellum and what could those changes do. And so we'll talk a little bit about how that sort of works for us. As a brief disclosure, this is off-label investigational use, and I don't have any commercial relationships, although you can change that yourself. Just send me some money and it'll all work out. So the way we really look at this is the bridge between Cambridge and Boston. So Cambridge on this side is home to MIT, where we consider like basic science and how do you sort of look at sort of the development of technology and tools to do modulation, and how do you sort of change the brain. And on the other side, in Boston is where all the hospitals are. So Harvard Medical School is over there primarily, most of the hospitals associated with Harvard Medical School over there. And that's where we want to do a lot of the treatments. And the way that we've often thought about this is this idea of doing clinical trials that sort of evaluate the treatment. And the way we look at this is that this is a huge gap, because we don't really have a jump between jumping into a clinical trial without a whole lot of basic science understanding. It's sort of like a huge translational gap that we sort of see. In other words, what we need to be doing is studies that can sort of link to treatment, but not necessarily assess efficacy. And studies that sort of are informed by the basic science and that with the goal of trying to get towards treatment. So a lot of what I'll be talking about today is our approaches to do this. What I'll be talking about is functional connectivity, just like Joel was talking about earlier. And one thing that's helpful to think about when you think about functional connectivity is it's really just looking at signals over time. So this is two networks, the dorsal attention and default network. Over time, and when you look at their signals over time, you can see that they sort of fluctuate in and out, and we can look at the relationship between these signals as a predictor of various things. So for example, here's a subject that has a high correlation between these regions, and this temporal correlation, this sort of temporal correlation that we look at sort of condenses out temporal information. So it just looks at correlations over time. But we think about that as a measure of connectivity, whereas other participants might have weaker connectivity, and this is sort of usually associated with worse clinical outcomes. The goal for today is to talk a little bit about our schizophrenia projects that we've been doing with Roscoe Brady. One of the points of today's talk is to talk about the negative symptoms of schizophrenia, which is a huge unmet need for patients. It's hard to treat them with medication. They're most related to the disabilities that we have, and they really hugely impair patients' ability to work. And so this is really the challenge of psychiatry is, we have antipsychotics that can treat the hallucinations, but we don't have these same antipsychotics are not really effective for impacting the other deficits that come with schizophrenia, and these are the ones that are fundamentally challenging for the patients. So using functional connectivity, what we've done is we've looked at a whole network-wide connectome association to sort of identify regions that are functionally connected and related to negative symptoms. And when we did this analysis and across subjects, what we found with 44 subjects, we found this region in dorsolateral prefrontal cortex and another region that I'll talk about in a second. And this region seems to relate to varies with connectivity as it relates to negative symptoms. So as symptoms worsen, something about the connectivity of this region changes. And when you look at it, this is what happens. These are the regions that sort of are changing with connectivity. You can see there that we have some regions in the cerebellum, and we also see some cortical regions here in blue that are reducing it, that are showing reduced connectivity as symptoms worsen. And if we look at how that really, what that really looks like, you can sort of see this network topography of here's a region that sort of has this particular connectivity normally, and then as symptoms worsen, this connectivity is going away. So we call this a disconnectivity hypothesis for the schizophrenia, where we think about these regions that are sort of becoming disconnected as symptoms are worsening. So the question really is, can you modify it? And so we started this almost a decade ago. It's kind of frustrating to think about this. I was talking to Mark the other day, and we've been doing this stuff for a really long time, where we were asking the question, can you change functional connectivity? So without looking at just the patients, first we want to look at sort of healthy human participants and see if you can really even move this connectivity. The way we did it is we would do a baseline stimulation session, baseline fMRI on the first day, sort of identify their brain networks that we wanted to target, very, very similar to how Joel was describing his work. And then on the follow-up day, we would do the TMS modulation, where we would do scanning before and after TMS, and we would look and see how that modulation has changed functional connectivity. And we do all of this right next to the scanner so that we can see how that would change. So the one piece of the story that I think is really important to sort of emphasize is that, and also directly after TMS, what you see is sort of no real dramatic change in the organization of these networks. I think it's important to recognize that we're not anticipating that like left inferior parietal lobe regions that are normally connected to the default network are going to somehow move to sort of into frontal regions or something like that. So the structure of the brain stays relatively constant, but the functional connectivity may change. So the organization is still present within the default network. So if you look at those change measures, then you can identify the regions that are changing. What we saw was that high frequency TMS, so 20 Hertz TMS, reduced functional connectivity in the default network. We saw low frequency TMS increase functional connectivity in this default network. This was really cool because we thought we were kind of onto something, and one of the cool things about this kind of work is that sometimes you'll watch the field sort of do these same things, and you'll start to see the patterns and the trends. And so this is something that I think Joel really sort of spoke pretty coolly about. That we see the same pattern, that the things that seem to be excitatory seem to result in decreased functional connectivity, and the TMS that seems to be inhibitory locally seem to increase functional connectivity. So we weren't the only ones to see this pattern. So Nabi showed this, and like I said, Joel showed it in his work, and we're seeing this again now in a new preprint from the Monash group. So what I think that there is this likely principle in cortical stimulation, where excitatory TMS protocols are going to reduce the connectivity and inhibitory TMS protocols are going to increase that connectivity. I don't necessarily know if this is the full truth, but I do think that this is a pattern that that's starting to emerge in the field. And I'm not the only one who thinks this, as you can see with that review article there. But there's a huge caveat to this, and that is like, how are you measuring this connectivity? This was sort of brought up in one of the questions before. Like, how do we measure this connectivity? Do we look between networks? Do we look within networks? And what kind of methods are you using are going to strongly influence how you look at the change in connectivity and whether or not that matters. And then the most important thing about this, relevant to today's talk, is that not all these targets are going to be the same. So the cerebellum is not the same. Cerebellum is definitely not the same as cortex, and not all networks are going to be made the same. So as we start thinking about where are we hitting and what are we doing, we need to be aware of that. As I've suggested, today's talk, we're talking about the cerebellum. We'll probably limit most of our analysis to regions that are sort of this anti-relationship between dorsal attention network and default mode network that are present within the cerebellum. So the point here is, if we really want to target the cerebellum circuit, we really want to drive this for schizophrenia, we need to do some studies to understand, is it even possible to do this, right? So if we apply stimulation to the cerebellum, how are you affecting this sort of cortical cerebellar loop? Are you going to be able to drive it through a polysynaptic connection to cortex? And is that even possible? And some of the challenges that we're still facing, even though we do know the sort of general principle that of course it's going to work, is that we don't really know how do we properly target the proper networks, and how do we properly dose it? And so this is what we've been thinking about, like, would it work? And then does it work, right? So how would we sort of do the modeling to predict whether or not it would work? And then how do we establish that it does work? So with Moritz, who's in the chat here, we've been looking at modeling of the cerebellar stimulation. So we did a systematic investigation of 150 locations in the back of the head to sort of look and see, does the sort of pattern of stimulation, how does that affect different networks in the cerebellum? When you go through and you do this systematic modeling, you can generate, in this case, E-field models of the impact on the cerebellum, and then combine these with the sort of cortical cerebellar functional connectivity networks to ask the question, what does it look like for coil positioning for targeting a given network? Where's the best place to stick the coil? Where's the worst place to stick the coil? And so here we're saying is that one location that might be really good for a cerebellum, for targeting, for placing the TMS coil for targeting the default network. Of course, you can look across a whole bunch of different networks and sort of find these idealized locations. And so this is the ideal way to look at it, where we can sort of see that there's some networks that show some strong specificity and some networks that don't, right? Some other crazy stuff that happens is some of these measures that we look at from cerebellum, classically thought to be motor, it's really hard to hit the motor structures here. And one of the things that I'm showing you is on the bottom there, this is the relationship between hitting the deep nuclei versus hitting the motor cerebellar cortex. And we can see that at no location is there pink, which would indicate that you're hitting the motor cerebellar cortex. Almost all of those locations in the back of the head seem to be influencing the deep nuclei, which I think is particularly fascinating. And of course, these predictions actually match really old literature. So if we map these locations onto the locations that have been shown to show inhibition, we see that there's a strong linkage between these predictions that we can generate. So it's really cool because we think that there's idealized coil positions to target a given network node. We can target this network, the green network here, or we could target the red network here just by moving the coil around in the back of the head. The other challenge that we face is the cerebellar dosing, right? So one of the huge issues that we particularly have right now is that we know that the cerebellum is different. We know that we need to do something to hit the cerebellum, but the choices that we make as a field have been really problematic, right? We've looked at a bunch of different ways to manipulate the intensity of the stimulation. As one simple example, as was sort of talked about earlier today and also yesterday. We know that you need to do something different here, but we don't know what. Should you measure the resting motor threshold? Should you measure the active motor threshold? Should you just not care? Should you calculate the distances? And so there's this huge challenge if you start reading the literature on cerebellar neuromodulation, because it could be that a lot of people have understimulated or overstimulated and cause a bunch of differential effects as a result of those stimulation attempts. And I'll just point out that I'm obviously still part of the problem. It's not just me. It's not just the field's problem. It's me and the field. Right? But the obvious question is, okay, so if you've done some simulations and you've looked at this and maybe you can properly attempt the dosing, like, is this even going to work? And we've invested a serious amount of time looking at this. So the iteration of this that we did was instead of doing this sort of baseline neuromodulation, sort of pre-scanning, and then targeting cortex, right? We did the same approach, but now targeting the cerebellum and looking to see if we could modulate network connectivity via the cerebellum. And as I alluded to, we can look at what we call on-target effects and off-target effects, and we can also look at the changes that we see from the cerebellum to cortex and also the cortical-cortical connections within the targeted network. So when you're on target, we see an increase in functional connectivity, both from the site of stimulation to cortex, But also we see an increase in connectivity within the cortical functional connectivity, suggesting that this sort of, you have this polysynaptic post-TMS response that sort of really could drive behavior. If you move the coil over now, and now you stimulate the midline, and you're no longer stimulating this targeted location, and we look at the functional connectivity from that previous target, and this off-target location, we see that there's no change in functional connectivity, either in cerebellar to cortex or cortical connectivity. And then when we apply sham, we see the same lack of change that we wanted to see in that network. Of course, because we did move that coil, there is a network that's associated with that from our modeling. We can see that it's probably going to be this dorsal attention network. So we do see this sort of reversal of network effects. Depending on where you are in the back of the head, you can hit different networks and drive different networks up and down. So as I suggested before, we also want to talk about stimulation intensity as an issue. So we've been doing a stimulation intensity experiment for the past couple years, where we've looked at three different stimulation intensities to see if we can drive connectivity and generate some data to sort of do some actual predicting of what the proper dose would be. We're still in the preliminary stages of this, of this analysis, but we see that we were successfully able to sort of do a low, medium, and high intensity stimulation. And if you look across all of our sessions, we sort of really spread out the sort of total stimulation intensities, so that we can now assess whether or not it has to do with the sort of relationship to motor cortex, or if it has to do with the relationship to the whole of the stimulator itself. First thing to say is that we were able to show that we can change the functional connectivity on a dose-related way, depending on whether or not you have lower high intensity stimulation. And that we see that as we look then to cortex, whether or not we're able to impact attentional performance, which was our sort of outcome measure along with functional connectivity, we see that what you really need to have happen is connectivity change to generate that attentional performance change. And this is something that I think Joel did really eloquently, to say, look, you need to have the connectivity change. It's not just enough to calculate whether or not you have the impact locally. It's more important to sort of have the whole connectivity pattern changed to better match what you need behaviorally. So what we think is that we can target it, we can engage that network, and if you do that, then you should see some clinical improvement. And so that's where we finally go, right? And so as I talked about, we have this sort of overarching theme. We have the schizophrenia patients that we know have this deficit that we think we might be able to correct with TMS. We've now been able to look at whether or not we can modulate these networks. So that's the fundamental question. Increasing connectivity via the cerebellar stimulation should happen. This network is deficient. Can we TMS this particular location and impact network connectivity? And so in order to do that, now instead of doing a single session, we sort of do a sort of compressed treatment regimen where twice daily TMS, and we do a resting state before, now 1 week follow-up. At 1 week follow-up, to ask the question, does functional connectivity change before to after TMS? And what we see is that we were able to rescue this connectivity function, right? So it was as you increase connectivity here, right, that results in symptom improvement. When you're failing to increase connectivity, we don't see symptom improvement, particularly in the sham group, right? And so what we're suggesting is that if we have the circuit down and the neuromodulation down, now the next step is to sort of have the intensity of stimulation and everything line up with the sort of intended connectivity goal. Of course, it could be that what we're really doing is not having a sort of focal neuromodulation, but we're having a sort of whole network effect, because again, the sort of analysis that sort of led to this sort of showed brain regions across the whole brain, right? And so it could be that really what we're doing is we're watching a whole network sort of track with illness, and that the TMS itself doesn't really cause change. The other option is that really we're seeing target-specific engagement of the connectivity of this sort of cortical cerebellar circuitry that we think is related to this disease. So in order to test that, we just did a whole brain analysis, asked the question, dorsolateral prefrontal cortex, where else are you seeing change? If we were sort of seeing the whole network, we should see this tracking, and it wouldn't really sort of be indicative of the TMS, but we do see that it's focal to the TMS that we did. So the only place that we see any change in the sort of targeted cerebellar targeted dorsolateral prefrontal cortex cerebellar network is in the cerebellum. This, plus a bunch of other data that I don't have time to talk about, makes us think that really what's happening, particularly in schizophrenia, is that there's this healthier state where you have a strong connectivity through the thalamus to the cortex of the cerebellum, and that as you become more ill, you remove that connectivity, and that because we've removed that connectivity, what you need to do is restore that connectivity to get patients back into a more healthy state, particularly given a specific symptom circuit domain. So hopefully what I've been able to talk to you a little bit about is how we've been thinking about whether or not we can model the target engagement, how we've been able to sort of suggest that this engagement of cerebellar cognitive circuitry is possible, and we can sort of measure its amplitude and predict it, right? And I've been hope I've been able to show you that if we do understand that sort of model, that we could potentially therefore engage this cerebellar network and cause change for patients. And we do think that that leads to a sort of unique hypothesis related to sort of a specific disease. Of course, I wasn't able to do this all by myself. I've been doing this for a really long time with a bunch of other people throughout the whole Boston area community. I sort of restricted some of the talk today so that we wouldn't talk about the cerebellar attention work that we've been doing, but that's also related to how we've been doing. Mark Eldaev, Roscoe Brady, Simon Lagonier have been extremely instrumental to this work, and Moritz has been instrumental to the modeling that we've done. So I thank you very much for your attention.
Thank you for the exciting talk. Let's jump to the questions. First, what kind of TMS coils do you use for cerebellar TMS to ensure focality? And how do you account for individual anatomical differences?
Right, so those are really great questions. So I think there's sort of two components to that question. So I think I'll start with the first. What coil are we using? We're using a regular figure of 8 coil. One thing that's important to recognize is that the sort of association cortex connectivity is linked directly to more superficial regions of the cerebellum. Almost all of the motor cortex stimulation is really trying to hit the cerebellum components that are really deep and are closer to the brainstem. And so that's why those studies have emphasized the use of double cone coils to try and reach those regions. But I do think that the figure of 8 coil is sufficient according to our sort of hypothetical modeling that we've been doing. The other question was how do you sort of accommodate the various changes in cortical structures for the individual participants? And the way that we do this, and the way that Joel does it as well, is that we do functional connectivity beforehand, and we sort of isolate the regions that we think are responsible for the sort of targeted network that we're trying to hit. I think this is a critical component. You need to be targeting the specific network that you want. There's an additional component to that kind of analysis, that is that you need to understand the sort of systematic organization of the brain, so that you're not just sort of picking some random hotspot that's all over the brain. You want to be picking the places that sort of correspond to the brain organization that you were intended to stimulate. And so that's what we do. We do functional connectivity, and we line it up with sort of network topography that's expected, and then target those locations. Given that you can do that, it seems silly to not.
Thank you. Let's follow with networks. The question is, when you stimulate different cerebellar areas that are part of different networks, like DAN or DMN, will the effects be different from stimulating the cortical parts of these networks? In other words, does the cerebellar node is just another node of the network, or it has some special value?
That's a really good question. We don't really know per se. That is something that I have requested funding for, but was that actual component was the component that was shot down. So that's a great question. We're investigating that in schizophrenia right now, so we should find out in a couple of years whether or not the trial is efficacious to cortical stimulation. I personally expect that it's going to be different because we know that the structure of the cerebellum and the frequencies of the cerebellum are dramatically different. One of the things that I like to sort of emphasize to anybody trying to sort of look at this kind of process is that it's unlikely that the frequencies of stimulation that we're doing, particularly in the cerebellum, are actually aligned to the rhythms. And so rules that sort of seem to work in cortex probably don't work in the cerebellum, and we're just putting noise into the system. However, in our case, even in that model, if you're sort of putting noise into the system and sort of causing the cerebellum to sort of jumpstart its working process, that's still okay and results in sort of a long-term change.
Okay, let's continue with the brain states. Do you apply TMS during the resting state? And if so, how do you think that task state would change the effects?
Great question. I think it will dramatically change. I think it will dramatically change, and that's something that I hope other people are interested in doing. We've been looking at resting state. We've been looking at resting state application of TMS. You know, again, theta burst protocols like this are relatively brief, couple minutes at most. So it's really hard to engage that sort of continuously to look for that long-term change. But I do think that given that what we know about brain networks and cognition, it's a pretty wise approach would be to sort of examine how those different states are going to impact the sort of network-wide changes. So the more that people are doing that, the better. And the more you want to let me know that you've done it, reach out to me, tell me. It would be great to know.
Let's hope you will continue this work. And another question, have you tried different modalities of brain stimulation like TDCS? Do you know something about that?
Have I tried it? Yes. We have a number of different modality experiments going on right now that I'm working with. My experience has been relatively limited. I think one of the challenges with all of these other modalities, particularly as we move into sort of a treatment domain, is these modalities that do not directly induce neuronal firing, sort of add an additional level of component of how do you sort of control for the task state, as the previous question was sort of working on. And so as we look at those types of questions, like TDCS or more complicated high frequency stimulation, we might have to think a little bit more strongly about what is the ongoing activity as we want to sort of modulate those.
And the final question, what are the best setup and participant position for cerebral stimulation experiments? Any special secrets about participant posture that may affect the outcome?
Yeah, so that's a great question. You know, we do head down seated because any other position does not allow as much access to the cerebellum. It does make neuronavigation a little bit trickier because you have to make sure that the coil, the coils that you're tracking, the subject's head that you're tracking, or all those markers are visible to the cameras that are tracking. So there is always a little bit of finagling of those positions before one does it. But once you do that, then it's relatively painless to sort of engage. The Cerebellar trial had participants hold the pillow and sort of put their face in the pillow and make sure that we don't disrupt the sort of neuronavigation tracking system. As one approach. The later attention studies just had healthy participants put their heads down in a chair. I would say in general, people have always asked me whether or not cerebellar stimulation is less tolerable. And I think they're thinking about a lot of these older studies looking at the double cone coils at really high intensities. And they're not thinking about what we're doing, which is a figure of 8 coil at sort of short, sort of theta burst protocols, which means that your participant is really sitting in the less comfortable position for a minute or two. And they actually feel like they tolerate those way better than when we were doing 28 minutes of 20 hertz stimulation to parietal cortex, which participants complain more about than they do the cerebellum.